My name is Oluwase Yolawale, and founder and CEO of Genoma Codings. I'm going to be sharing you a lot of secrets as touching unlocking the secrets of plants. Okay, secrets on unlocking the secrets, because whatever remains a secret becomes a mystery, right? Right. So, and if you want to unlock the mystery, you have to share the secrets. Am I right? So, I'll be working with you and be presenting on exploring the future of plant science and genetics, where we are going to look into how to unlock plant identity with DNA barcoding techniques. I repeat, we're going to look into how we can actually be able to unlock plant identity with DNA barcoding techniques. So it means that in the course of this webinar, we're going to be looking at possible ways on how we can use DNA barcoding to revolutionize plant identification and conservation efforts. Okay, we're going to be seeing how we can use barcoding techniques or understanding barcoding, all right, in the course of this webinar, to see how we can revolutionize plant identification and conservation efforts. You see, plant identification is key. I remember there was some information. We get to understand that over time, right, identification of plants in the old days have to do with, a kind of a generational or cultural traditional a thing whereby this knowledge is being passed across from generations to generations. You get to discover that this person has to take the child. I was saying that identification of plants in the old days have to be done from, passing out knowledge to the next generation, and to the next generation and to the next generation. And so far, nowadays, when you go to maybe to the farm to identify plant, there's still some plant that gets you confused, right? And at the same time, we don't want to make mistake to get plants that leads to poisonous and some other related things. There are quite a number of different sets of research that you really want to carry out, right? That you really want to do, you want to carry out in the course of your research purposes. But identification of those plants and to know that these plants are really doing what they are doing, and also to be able to even identify the ingredient and be relating it, related to the course of your research and objectives of your research is important. And that is why these particular techniques have come to play, right? To see that you are equipped with the knowledge and the skills and capability to be able to see that you can easily identify plants using DNA barcoding. There are other techniques that we're also going to be sharing. I'm also going to be talking about. And at the end of the day, because today is not enough to actually know how to do these things. So I'll tell you the way forward on us being able to do this, probably after this webinar. Okay. So understanding DNA barcoding in plants. Understanding DNA barcoding in plant. The significance of DNA barcoding for plant identification is crucial for accurately identifying plant species, right? The significance of DNA barcoding is crucial for accurately identifying plant species. So you could just say that DNA barcoding is a technique used in identifying plant species. And also it gives you the ability to be able to enhance biodiversity conservation and understanding ecosystems. So when we talk about conservation, we're talking about, what's the first thing that comes to you when we talk about conservation? Yeah, can somebody just drop that quickly on the chat room? What's the first thing that comes to your mind when you hear plant conservation? Just a word at the chat room. Just a word at the chat room. Anybody? Don't unmute yourself, please. Just a word. Yeah, somebody said nature. Nature. Yeah, very good. We want to conserve nature. The first word that comes to what's your name? To Fatihat mind is nature. Okay. Mubarak Z Adesola says keeping safe. Marvin says protection. Right. AY said to avoid extinction. Basse said protection. Yeah. Another one said preservation. Amazing. Another one say preserve, another one say plant protection. Exactly. You are all correct. When it comes to conservation, do you know that there are some plants that are going to extinction? I was expecting somebody saying extinction, but I think another word. If you don't use okay, yeah, I think I saw. So AY says to avoid extinction. Exactly. To avoid extinction is to preserve. And the process of preserving, as far as plant-related studies concerned, is conservation, right? Because there are some highly medicinal plants. There are some plants that are so nutritious. There are some plants, do you know that some plants have been lost because of there's no enough archive to store them, or some information has been lost due to fire, and so many other things have been lost out there. There are some plants that are not poisonous, but till today people still think that they are poisonous. Are you getting my point? There are plants that have the natural capabilities to be able to solve a whole lot of health pressing related issues in this world. It was said that over 40% of the world medicine, right, is gotten from plants. Who can tell me one of the medicine you know that was discovered from plants? Just give me one of the name of the medicine very quickly. Not from microorganisms, okay? From plants. Anybody? Because this webinar is a kind of a form of discussion, right? We have to deliberate and converse, right, around it. Yeah, somebody say artemisinin, right, to deal with malaria. Yes, very true. And there's a story around I think artemisinin. I hope it's artemisinin. How that this woman was the one that discovered it, and up to today, do you know the family of that woman is not getting royalty? Am I right? Can anybody confirm that? I believe it's artemisinin. Artemita, right? Yeah, somebody say yes. Yeah, AY said yes. Yes. Up to today, the woman that discovered artemisinin, the generation, even the woman and the generation of that woman has not gotten any royalty. Are you getting it? And that's an issue. That's an issue, right? So there are so many drugs, there's so many diseases out there that by nature, they have the capability to be able to address those issues. And I want to tell you that truly, research has not gotten to that point. But I believe there's a plant that can deal with HIV, and there's a plant that can deal with cancer. I mean, that can solve cancer. It's just that in the current state, or let me say, in the contemporary age, a lot of medicinal research has gone to, more of compound-related combination sort of things and synthetic stuff, rather than being able to investigate deeply into the nature. And I want to say that that's the future of medicine. You begin to discover that there are new revolution when it comes to startup, when it comes to a solution-driven initiative that is coming around using nature to address related issues. Because the truth of the matter is, there are inbuilt related nutrients and medicinal properties that can even deal with aging. Do you know that? That can deal with aging. Am I right? Can somebody confirm that? When we talk about aging, haven't you seen some people who I mean, those, our great grandfathers that all of their life is nature, and you can discover that they lived long. Am I right? Can somebody confirm that with me? Yeah, exactly. And you get discovered that the contemporary drugs, they say, oh, there are some drugs that they are without, and they have side effects, and even the side effect is causing another issue. Yeah, so there's a lot of potential, there's a lot of result, there's a lot of things we can do with nature, with plants, right? And quite a number of different things that we can use plant for. And you get discovered that even our feeding and nutrition is as a result of plant-related, nutritional foods around us, right? As a result of plants. And you get discovered that these plants, or that this plant, they don't have side effect, because if we keep taking food, we take vegetables, we take fruits, and none of these things have side effects. It means that there are some plants that we could actually leverage to address a lot of biological related problems. So this DNA barcoding still has to do with innovative solution, addressing innovative solutions, being able to discover innovative solutions. So if you are an innovative driven person, all right, if you are an innovative driven person, it means that this particular platform, this particular techniques is something you really, really want to consider, especially somebody that is diving into or has interest in data science, that has interest in machine learning. There are a lot of DNA barcoding data that you can gather to be able to come up with a specific tool that could enable you to develop innovative solutions that can quickly identify plants even without going through molecular characterization and identification techniques. So many innovative innovative people here, people who want to dive into not just research for publication purposes, but also to be able to like come up with an innovative solutions that could maybe integrated into tech, right, to address quite a number of different challenges out there. DNA barcoding data is a very important data that has not really been explored, especially in the area of data science that we could actually explore in, especially for research in PhD and all, right? So it's very important. Like for quick DNA identification, it's key, all right? It's very, very key. Sorry, I'm going to be sharing my slide back shortly, all right? I'm sharing back my slide. Please give me a minute. I believe it's the network. Please, I believe you should be able to see my screen now. Am I right? You can just reply to the chat room with a yes. Just reply at the chat room with a yes. Amazing, amazing. Thank you so much. Yeah, so DNA barcoding is crucial for accurately identifying plant species. I've said that before. Enhancing biodiversity conservation and understanding ecosystems. So it allows scientists to confirm species authenticity, essential for medicinal plant verification and preserving endangered species in our changing environment. There's various numbers of patents that could come as a result of you leveraging on DNA barcoding, and a lot of bioinformatics tools that you could also use to, you could actually develop in the computational biology part area to be able to do that. But there are people who are just, who are researchers, right, in the academic space and would like to use DNA barcoding in their research for DNA plant identification using some specific motifs, right? There's some DNA data, sequence data that we're going to get, the repeaters, motifs that we're going to walk around in the next couple of days, okay? By the time we are through with this webinar and you get onboarded. So we're going to be using some set of tools to do hands-on practical in addressing this particular issue, okay? And also, we're going to be talking about some innovative areas you could actually use DNA barcoding to come up with various number of solutions, but just by incorporating data science and other related tech concepts. Okay. So applications of DNA barcoding, we have verifying plants, ensuring authenticity of what medical plant species. So we have DNA barcoding helps to confirm the identity of plants. So there's something generally in genomics, right? And that is the place of maybe like after you done PCR and you do ITS sequencing, like target sequencing data, right? So there's something we call molecular characterization and identification, where you use some set of online tools, online bioinformatics tools to identify those plants. Now that is first thing, molecular identification, okay? Now there's still also the DNA barcoding part, right? Which would really help in you being able to identify some of these plants. I think the scenario still comes around you being a researcher, you can just like explore generally plants out there and get as many plants as possible. And I think there was a platform in Africa called Africa Plants, is it Africa Plants or Animal Biodiversity Project, right? Initiative. How many of you know that? African Plants Biodiversity Project. So it's a set of long years project to identify plants in Africa, like as many plants that has gone into extinction, some novel plants. So you want to be someone so novel in your research. I think this DNA barcoding is also important because it enables. There are a lot of plants out there. In fact, there are various number of plants that has not been identified yet. Do you believe that? Please, can I get a confirmation to that? Or do you think scientists have discovered all plants? Yeah? Have scientists discovered all plants? Yes or no? So, and if no, that means yes. There are lots of plants out there that are yet to be discovered, and the fact that more plants has not yet been discovered shows that there's a potentiality of discovering novel plants that can address world pressing related challenges, especially outbreak infectious diseases and quite a number of different diseases out there. And you see, in the concept of innovation, there's something we call multi-omics research. I repeat, there's something we call multi-omics research. You can bring in the concept of multi-omics research into what we are talking about today, into plant research, whereby you use DNA barcoding to discover novel plants out there that has not been discovered before. Are you getting my point? Yeah, somebody says a lot of plants are yet to be discovered. Very, very true. I agree with you. A whole lot of plants has not been discovered out there. So the use of DNA barcoding enable you to discover, to also be able to identify a novel plant that has not been discovered before. Because you see, this DNA barcoding has a database. And by the time we get some of these specific gene markers or specific motifs and repeaters within the sequences, and we blast, we are able to identify the plant. The database is able, the tool set, online tool set on the database is able to tell us what that plant is. But a plant that we could not identify begins to give you the concept that this plant could be novel.
Genomic. One second.
Continue. Yeah, so as I was saying, right, that you could actually discover a novel plant through using DNA barcoding and other molecular related techniques that you are going to learn in the coming days, okay? Through hands-on as well, okay? So after that, I was talking about multi-omics research concept, okay? So the multi-omics research concept enables you to be able to, from novel discovery, initiate those concepts that will enable you to discover potential novel properties within the plant that could address various numbers of problems. Okay, so multi-omics one begins from probably maybe when you do genomic sequencing, and then you take the genomic sequencing using superior number of therapeutic related tools to discover more of like secondary metabolites, okay, biosynthetic genes, novel biosynthetic genes through those tools. Okay, so first thing is genomics. You do sequencing, you do whole genome sequencing, you get all the entire genetic sequences that's relating to the structural and functional related capabilities of the plant, which encodes the entire DNA and proteins or enzymes that has to do with even possible medicinal related properties, okay? Like encoding possible medicinal related properties. That's at the genomic level. You do functional genomics, you understand all of these things, you're able to identify those genes, right? And then you now begin to take it to probably metabolite level, where you use some specific tools like anti-smash, right, for plants, and then you are able to like run some of these tool sets on bioinformatics tool set on the plant's genomic data to discover molecular, secondary metabolites, biosynthetic genes, and their pathways, right? And then after that, you now take it to probably using machine learning to see how you can redesign or re-engineer some of these secondary metabolites to become more of like peptides, and then from there, you do the synthesis, the biosynthesis of those peptides, and then you apply it on various number of diseases. You then take it to transcriptomics to see the gene expressions of whatever you're applying. You understand? And then, or maybe the peptide you can use drug discovery, molecular docking. So basically, some of these plants, nowadays we have an approach, right? There is a general approach, right? Yeah, there's a general approach where you just do like maybe extraction. Am I right? Is that not the general approach out there? Extraction, then you take it to identifying maybe I don't know. I don't do much of that, but it's a general way you just do extraction. Then it then leads you to drug discovery. But when you do molecular, there's kind of a sure area of being able to like streamline your research to a specific targeted gene. Are you with me? Or targeted protein that is responsible for specific medicinal properties, right? Targeted medicinal properties within the plant. Are you getting my point? And that is the multi-omics aspect. So multi-omics aspect is like a combination of infusing or integrating different omics studies, like genomics for DNA. Are you getting my point? Proteomics for proteomics, proteomics for protein study, transcriptomics for RNA, metabolomics for you understand. But different research concept or techniques or methodology between each of these areas can be infused and integrated for a workflow of discovery, right, to a specific point that you are needed. Presently, at Genomark, we are doing a nationwide research on Vibrio cholera, right? And We are doing like a multi-omics integrated research concept where we are combining both the functional genomics. We are doing comparative genomics. We are doing variant genomic study. So the comparative genomics do with a lot of evolutionary studies and diversity within the sequences. We have the functional genomics to identify genes, proteins, their functionalities, and every set of things that they're responsible for. Are you getting my point? Yeah, so and after that, we're now going to the therapeutic discovery where we're going to be working on plant-related data. So those plants, we are using different concepts to be able to see that we discover all of this therapeutic potential within them. Specific genes like secondary metabolite, biosynthetic genes within them that are redirected to a specific medicinal property we're looking to address a particular problem. And that is basically maybe to engulf this virulence genes or a specific drug target within the Vibrio cholera that we have, current Vibrio cholera that we've sequenced and all of that. So can you see it's a whole lot, right? But then everything still boils down to which of the novel plants do you really want to work on, right? Which of the novel plants do you want to explore? Which of the novel plants do you have you been able to discover? So DNA barcoding can play that crucial role, right? So we have conserving species like protecting endangered plants through accurate identification. So it plays a crucial role in conservation efforts for endangered species. Then we have improving crops, right? Enhancing agricultural practice for better yield. So this technology aids in developing superior crop varieties for farmers. Now, how does that play out, right? So using DNA barcoding to be able to identify, you can actually use it to identify varieties of crop species. Now, let me put it this way. You know, we have a particular crops, but over the years, this crop could evolve because of environmental factors. Are you getting my point? To different sort of varieties of that particular crop, right? Right. So, for instance, in rice now, you know, we know that we have rice, but there are different cultivars of rice and so on and so forth like that. So basically, variations play key area, and DNA barcoding also works with variations to be able to see that you are able to identify crops of the same species, but you know, like they vary on the basis of genetic variations and mutation within them. So there's a technique around that on how you can deal with that. Probably in the course of our hands-on program in a couple of days, we'll be able to address that also with some basic software tools, bioinformatics software tools to see into that as well. So probably we use, because we're going to be doing multiple sequence alignments, I could show you around calling for variants and being able to identify those variants around some specific motifs that would also serve as a DNA barcoding for identification. Yes. Somebody want to say something? Please. There will be time for questions and answer, okay? So where you unmute yourself and then we discuss, okay? So we have tools for DNA barcoding like NCBI, so National Center for Biotechnology Information. So an NCBI provides access to vast array of genetic data. So the genetic data is out there. So basically, when you don't have money to do sequencing, we could actually get some whole genome sequencing data or specific targeted sequence data to see that how you can use them to be able to get this data, and then use some other tool set like BOLD, Biocode of Live Database, which is specialized tools used in plant identification through DNA sequences. It's also used as a barcoding tool, all right, to identify those things. Then we have primary design, tool for primary design. Now, this primary design is specific. So you can use DNA barcoding to identify a species and then be able to come up with a specific motifs or genetic targeted sequence areas that are of interest. Then that will now lead you to primer blast, okay? Like primer blast, like, you know, being able to like, because there's a particular tool in NCBI that can do primer design, but there are other tools that can also do that. In a couple of days, I'm going to be showing that to you specifically on how you can design primer for plants for your your research purposes. Now, there's also a way where you can design primer for novel plants. That also has to do with incorporating both barcoding and primer tools together to be able to design specific regions that you need for sequencing in order to further your research in specific areas. Then we have DNA analysis, like interpreting results from DNA sequencing. So data analysis is essential for accurate plant identification and classification. So in the course of that, this DNA sequence analysis has to do with specific tools in bioinformatics tool sets to be able to do that. Now, some of this DNA analysis, after you've done like the barcoding and all of that, then you're able to get primer design for those areas. Like, basically, for instance, you have to get sequencings from NCBI. You use both to do some of those barcoding techniques. And then from whatever you get from there, you're able to like design the primer. You send it out for sequencing. The next set of thing is DNA analysis. Okay, DNA analysis. So this DNA analysis is essential, whereby you are able to accurately analyze the plants, not just to identify the plant, but identify specific genetic components or biomarkers that are related to your aims and objectives, and basically what you are researching on. Okay, so that's very essential. So there's really a number of tool sets that we can do, and this DNA analysis, that's where we begin to have like phylogenetic analysis, GWAS. How many of you have heard of GWAS? Genome-wide association study. We have transcriptomics for plants. We have variant analysis, detecting variants, and the implications of those variants when it comes to the biological functions and morphological behavior of the plant, especially those relating to stress resistance, disease resistance, crop productivity, yieldedness of the plants, and quite a number of different areas, right? Is very, very key. Then we have research collaboration, like sharing findings with the scientific community. So with this DNA barcoding, like some of this barcoding you are getting, and I mean, when it comes to the research collaboration area, you could like launch an initiative whereby you are able to build a database where there can be some sort of collaboration and open access data set for further research and expansion, right? Like progress. So collaboration enhances the understanding of plant biodiversity and conservation efforts. So there could be some initiative around that, and you could actually apply for grants to get some grants initiative in that. So step by step for plant identification, like what are the step-by-step approach to carry out this plant identification? We have understanding the process of DNA barcoding, which involves the use of extracting DNA. So first thing first, like I said, you need extraction of DNA. You need to amplify the region, right? And then you need to like sequence those regions. Okay, so that's like targeted sequencing, and you can do ITS sequencing and all of that. So especially in a low and middle income country, because of lack of funding, one of the things you could actually do is do targeted sequencing, which will not cost you a lot of money. And you can, through this targeted sequencing, take your research around evolution, okay, around evolutionary study, where you get the DNA sequences from public databases like NCBI from around the world, depending on different kind of plants you want to work with, and then be able to design a research problem and a research idea, research aims and objective around that specific plant. Okay, to look at evolution, because there's a lot evolution can do. Evolution came as a result of a lot of environmental issues, which maybe leads to genetic variation, because there's no evolution without genetic variation. The reason why we have evolution is because there's genetic variation. Are you getting my point? The reason why we have evolution in plants and in animals and or humans, basically, right, is because there are genetic changes. There are mutations that take place at the genetic level. So when you are trying to study specific areas in plants, or you explore a specific research area in plant, it is also important you consider evolution, because evolution could have a lot to do with why some of these challenges or issues arise. For instance, maybe you have a cocoa farm, you started this cocoa like 5 years ago, and discover that that particular varieties of cocoa you are using is like producing a whole lot, and it's a specific texture to your cocoa product. Are you getting my point? There's a texture to your cocoa product. Now, over time, you begin to see that there's like a degradation, like in the texture of the cocoa product, and even in the productivity of that particular cocoa. Even after changing land, you still discover that the productivity of that cocoa is not really yielding as supposed to be. By the time you begin to investigate, like you can take your research to that level where you are able to like investigate and take the matter to the genetic level. There you begin to see insight into specific genetic mutation, variation that has occurred probably as maybe a particular event that occurred one time, more maybe like erosion, more maybe like it has weather condition that could have made that plant to evolve and then be able to adapt that capability, and then you begin to discover it's affecting the productivity of the plant more than how it was supposed to be before. Are you getting my point? So these are like steps. So it's beyond just plant identification. It's beyond it. It's beyond it. It has to do with a whole lot, because you cannot eventually carry out a research on a plant you don't know. Am I right? Can you carry out a research on a plant you don't know? Right? you definitely have to identify the plant. So all of the things I'm saying are like further areas of research you could actually carry out, but then everything starts from plant identification, especially when you want to carry out a novel adventure of plant discovery. Okay, and a lot of that. So, and then the data is then matched against databases to confirm the plant identity, ensure accurate clarification and verification. We have real-world research examples like impact of DNA barcoding on plant science. So DNA barcoding has significantly enhanced our understanding of plant diversity. Okay, so we have like research shows that it effectively identifies species, verifies the authenticity of the medicinal plants, and aids in the conservation of endangered species, ensuring biodiversity preservation. Can you see that? Hello, please, can you hear me clearly now? Somebody says my voice is not clear. You can just reply with a yes. Is it clear? Yes, amazing. Thank you so much. So it effectively identifies species, right? Verifies the authenticity of the medicinal plant and aids in the conservation of what endangered species, ensuring biodiversity preservation. Some of you who are so much interested in conservation, preservation of plants and all of those things, DNA barcoding is a very, very powerful tool for that. Okay, very, very powerful tool. So some of the things it does is like spot fake plants, protect endangered species, and then boosting crop development, right? So that's it basically. This is just like an open session to understand what all of this thing is all about. We're going to be having a workshop. Let me just quickly share that with you. Please, can you see my screen? Can you see my screen clearly? So we definitely can actually not be able to do all of these things intensively, right? We cannot do all of this intensively without a hands-on session, and it cannot be done in one day. I repeat, it cannot be done in one day. Okay, so if you really want to decode nature and then be able to like do all sorts of these kinds of analysis, we have to give it like at least 2 days to be able to do that. So we have decode nature, which is primary. It has to involve or it's involving primary design. Okay, we're going to be using various number of tool sets that you can use to design primers on specific specific plants that you really want to work on. Okay, so and then you'll be equipped with that, with the capability to be able to like design them using different set of tool sets, even paid tools like some set of bioinformatics software tools that are paid, but you are going to get it for free in the course of this session. Okay, please, can you see my screen? You can just reply with a yes. Just reply with a yes. Amazing. So we have primary design and DNA barcoding analysis for plant species and education and intensity. So it's a 2-day hands-on short course. I repeat, it's a 2 days hands-on short course. And what is this course about? So you are going to learn how to truly identify plants using DNA, not just leaves or flowers. Okay, so the traditional one is just using leaves and flowers to be able to identify some plants. So this hands-on short course empower you with the in-demand skills in DNA barcoding, multi-platform species identity, primary design using tools like NCBI, Board, DNA Subway. We're going to also take you to cluster, omega, primary design, and possibly I will be taking you through some variant study. Okay, from the cluster omega, I will show you how you can identify some variations and to study around evolution that could arise as a result of various number of factors that could address specific biological conditions, or affect specific biological functions or conditions, or relate to specific biological issues that you may want to address in the course of your research objective. Okay, so you will also design your own DNA barcoding strategy, retrieve real-time plant sequences, validate them using multiple tools, and build PCR primers, all without stepping into a lab. Okay, so what you will learn in the course of this 2 days hands-on session is how DNA barcoding works and how it's changing plant science. And so we're going to explore more. Then how to, we already say much of it. So how to work with, and how to retrieve DNA sequences and validate species identity, how to compare data across platforms, and then basis of PCR to design accurate primers. Then we have real-world applications about medicine, agriculture, biodiversity, and diagnosis. So are you getting my point? And then we're also going to look at those who have ideas of plant science, like, data science, area where you want to dive into ML, machine learning, how you can gather some of these tools to be able to build groundbreaking tools, right? We could share some of those areas, like into some of those areas and ideas to come up with groundbreaking tools that could be like an app where you just like use that app to use it to identify plants very quickly, right? It's very important. Yes. We're going to share the slide, okay? We're going to share the slide. So hands-on tools you will use, like we have NCBI Gene Bank, both system, DNA Subway, Cluster W, Mega Eugene, Primer Blast. So these are the things, the people that should do are definitely you, students in both biology, botany, agriculture, biotechnology, herbal product innovators and researchers, conservationists, and then biodiversity scientists, educators, and nature enthusiasts, okay? So this is the curriculum. We have introduction to DNA barcoding, and these are the activities like barcode theory, gene regions to identify. We're going to dive much deep into that. We're going to look into study design, like choosing the right barcode region, NCBI sequence retrieval, like faster download, metadata logging. We have blast analysis. We have multi-platform analysis. We have alignment view. And this alignment view, I'll be walking you through evolutionary study as well. We have PCR theory, primer design, group presentations. And what you will get is like certificate of completion, plant barcode, and primer design toolkit. You're going to get, you're going to go understand sequence retrieval and login template. You're going to get sequence retrieval and login template, assess G-Nature's follow-up project, mentorship, and guided mini research invitations as well will take place. So it means that that's some of the bonus you get. So, and this is why it matters. It matters because accurate plant application herbal validation. For those of you who have so much interest in herbal validation, especially you're working in institutions that are really working on herbal innovative products development, you need this skill. Then we have crop protection and species conservation. This course gives you practical bioinformatics experience that supports both research and real-world innovation. So it's like 2 days. It's not on site, please. It's virtual, okay? It's virtual. We're going to go from beginner to intermediate level. You're going to get certification as well. So that's it. This particular program, I think this course is coming up. Is it next week? I think it's coming up next week. More information will also be dropped at the chat room. But before I continue, do we have any question at all? Do we have any question? Do we have any question? How many of you have Genius software? Do we have anybody that has Genius software? Anybody with Genius software?
No, sir.
Do you know Genius software? We're going to be using Genius software in the course of this training as well. And this Genius software costs. Okay, somebody has Genius software. Do we have anybody that has Genius software? So cost for Genius software is around $200. Okay, cost for Genius software. It's a bioinformatics software to do quite a number of different analysis. Okay, and then you need to get access to that software before the program commences. Can you see? So the price ranges from $4,500 to $22,500 annually. And you are going to get access to the lifetime activation key of a version of this software for life, okay? Like lifetime activation key. Can you see the range of this software? $4,500 to like $22,500 per year. But we have the lifetime activation key. For student version is around $200. Can you see that? Student version is around $200. I think this is the other one is like academic version. Is that okay? So, but you are going to get lifetime activation key for that in the course of the 2 days. Okay. So, no. It's not going to be free. All right. Adiola, it's not going to be free. Okay. It's not free. It's still paid. So I think I'm on the behalf of the organization, on behalf of you, I'm pleading to the organization to leave this particular session for $7. Okay. $7. Is that okay? For those who be interested, I think we should go with $7, especially with the software tools and other bonuses you're also going to get in the course of this program. Can you see? Can you see the users per year? These are the users per year, but we have the live activation key. We have the live activation key. This is for personal subscription, like $200. Can you see? This is for personal subscription. If you want to buy this software. Okay. So Olamileke says that it's too much. Well, I think that's what we can do. Wait for this webinar. Meanwhile, there's going to be certificates for the hands-on program. Okay, there's going to be certificates. Yeah. So that's it basically. Any other question? Any other question? At least, if not because of the program, this software is very important because there's a various number of things this software can do, especially on our upcoming programs as well. Okay, you really need it. You really need it, especially when it comes to evolution and variant studies and all of that. This software, transcriptomics, you want to do predict open reading frame, do gene expression analysis and all of those things. This software can do that. This software can do that. So that's it basically. And we're going to be touching some other things beyond what I just showed you. By God's grace, I will be spending time to teach you in a couple of those 2 days. And after those 2 days, you'll be given a project to do, and then you present it. Is that okay? After those 2 days. So any other question? Okay, there should be certificate for today. I will let them know so that they can work on it. Okay. Yeah, the software can also work for human genome. Yes, definitely it can work for human genome. Definitely. Any other question? I'll be going now. I think we're done with our presentation. It's just like 1 hour. Can you use phone? Yeah, for the DNA barcoding for the workshop, you can see that some tools we are going to use beyond just the software. Okay, so definitely you can use your phone because of some other tools we can use for DNA barcoding analysis. Okay, you can use your phone as well. Yeah, it's the same software for bioinformatics as well, definitely. Okay, all details on the WhatsApp group we shared as well. Yes, no problem. Yeah, so that's it. Thank you all for joining. Looking forward to meeting you in our next webinar program coming up probably next month. Have a great time. Thank you all for staying with us. Thank you all for joining. And we look forward to making more impact with you all. Have a great time. Thank you. Over.
If you have any further questions, you could just type them in the chat box while I try to provide as appropriate answer as possible to everyone. To those who said that was the program advertised free, only this webinar was advertised free. The primer design course is not free. However, we'll be giving generous discount, like the CEO said. For those who have the genome, the software before now, and then you could always reach out. But I think $7 is just enough discount to be given. I want you to consider that. I'm sorry we're not providing certificate for this webinar, except there is further consideration from the director of the subsidiary. If you want to make an appeal on that, you can send an email to info@gnhs.com so that we can consider that. If there's any question I would like to answer before so the link to this today's webinar would be shared as via the email. So everyone who participated today will get the link in their email. So just look out for that so that you might be able to do a reward for what you have learned today.
Hello, sir.
Yes, I'm with you, Mr. Salisu.
Okay. Thank you for organizing this wonderful webinar. Sir, I am among those who already have the junior software license because I attended several of your webinars and even research intensive. So I already
Okay.
have the software and so is there anything that you can do for us?
I am not in a position to give a word concerning that, but what I would advise you to do, Mr. Salisu, is that please join the WhatsApp group and then you can reach out via the email I'm sending here, genomarknatures@gmail.com or info@gnatures.com. So,
Thank you.
yes, if there's anything we can do in that line, definitely it's going to be done when the director is on seat. I hope I have answered the question, Mr. Salisu.
Yes, sir. Yes, sir. Thank you, sir.
Thank you very much. Any other question, please? Yes, Mr. Daniel, the slides will definitely be shared alongside the link to rewatch the webinar on YouTube via the email. Thank you very much for joining us today. I look forward to seeing you on the inside. Even if you are still considering whether or not to take advantage of the primer design course, I would advise that you join the WhatsApp group and then you look forward to further information through the group. Thank you very much. Have a nice and a wonderful day.